Alright guys, so continuing on with mitosis, we're going to do the stages of mitosis, and again we're going to color code a little bit. I'm going to use pink and blue, but we're going to go kind of simply through this. It is kind of confusing to most, but I'm trying to simplify it for you, so if you have questions, please send me an email. So let's go ahead and get started. So the first step starts with an I, so I'm going to write that down here on the side. This is called interphase. So this is basically, I'm going through the steps of how we start with one cell and get two identical cells. So we call this the preparation stage. Okay, the preparation stage. The cell is not dividing yet, okay? It's just preparing to go through mitosis, but it's not dividing. I hope you guys can read my handwriting. This is where the cell is going to double in size so that it can prepare to separate. So they're going from like a very, very small, cells are tiny, but it's going from small to doubling so that it has space for the chromosomes. We have our DNA, our deoxyribonucleic acid, is duplicated. Or another word for that is doubled. Okay, so it's going to duplicate, okay, or double. So when we start, okay, we are going to have one cell, okay, and it's going to basically, we're going to have one blue and one pink. Okay, so these are the mother cell. So what's going to happen is the cell is going to double in size. The DNA is going to be duplicated. So within this interphase process, basically these chromatids are going to become chromosomes. So it's going to go like that. It's going to double. Get my colors mixed up here. Okay, so it's going to look like that. So that is what we call interphase. Okay, interphase. Then we're going to start going and doing the steps of mitosis. Okay, so mitosis, so interphase was the first part. So this is basically the prep before mitosis actually starts. Okay, so I'm actually going to underline this in a different color so it looks better. Alright, so then we get mitosis. This is where the cell starts the divide process. Okay, it has to make exact copies. So in mitosis, generally speaking, it's the nucleus starts to divide. Okay, then the chromosomes are going to condense. Okay, then we start getting our other phases. So we have P, M, A, and T. So we have ip-mat at this point. Okay, and remember you can always pause it to get caught up. So the first one is called prophase. Okay, now these four p-mat are part of the mitosis process. So the ip-mat is how we typically remember it, but prophase, okay, in short, is when the chromosomes, so these chromosomes here, push together. Okay, they push together. So prophase, they push together. Okay, another word for that is condense. Okay, they condense towards each other. So with that, okay, Okay, they basically condense in the middle. So not a whole bunch happens, they just get kind of squeezed together. Alright, M stands for metaphase. Okay, metaphase is where they line up in the middle. Okay, so the chromosomes, and I'm going to start abbreviating so that you guys don't have to write chromosomes 100 times. They line up in the middle, okay, which is kind of what's already happened, but they line up in like a straight line. So again, very similar thing. They just line up directly in the middle, straighten up a little bit. Then A is anaphase. Okay, anaphase is when it starts to actually separate. So this is where things get kind of interesting. So we get these things called chromatids, which if you remember is half of a chromosome. So the chromatids, half of a chromosome, are pulled to opposite sides of the cell. Okay, so this one, so P, push, M, middle, A, this one was a little harder to do, but they are pulled to the end. So this one, you're going to draw where it's starting to kind of form two cells like that, where it's going to divide, and you get one blue here, and one blue there, and half of the pink, and half of the pink. So they're starting to divide and go to the sides. T is what we call telophase. Okay, telophase is where the chromatids, again, half of a chromosome, so these little ones, not the X's, but just the little squiggly lines, the chromatids move to the ends of the two new cells. So they're just moving more towards the end. Okay, it's starting to divide a little bit more. So it's where they move to the end. So we have more of it's starting to actually divide. Okay, so we have, again, the blue and the pink. Okay, telophase. Alright, then our last part is a completely different step called cytokinesis. Okay, this is where it actually cuts into two. It's going to actually separate. So, in all of the steps together, it's ip-mat and then cytokinesis. This is where the cell officially divides into the two new identical daughter cells. Okay, that's what cytokinesis is. Okay, so I'm going to draw that out. So now you're going to have two completely new cells. So telophase is where they're starting to go away. Cytokinesis is when they are officially two new identical cells that look just like the start one. So one blue and one pink. Okay, so they have the same exact one. Now we would call this is two new diploid, which means it has the same DNA as the mother cell. Okay, diploid means Okay. All right. So I'm hoping at this point you guys understand mitosis a little bit better. It is a little bit more simple than the original one, or of meiosis, which is more sexual reproduction, which is going to be the next video. So I'm hoping that this makes sense. If you guys have questions, again, please email me. Have a good day.